Hey guys, I hope you all are doing well. Today we're going to do a new topic, that is absorption of drugs. Previously we have started this chapter, that is pharmacokinetics. Rest of the videos are present on my channel. If you haven't seen them, go watch them before watching this video so that all your concepts can be clear. Okay, and I recommend that you keep this book with you, and this book is Lippincott Pharmacology 9th edition. We're going to study from this and keep on highlighting the main important things which we study together. Okay, so mechanism of absorption of drugs from the GIT tract. There are basically 4 different types by which this drug can be absorbed. For example, passive diffusion. What is passive diffusion? It means the movement of drug from the region of higher concentration, as shown here in the extracellular space, to a region of lower concentration, that is the cytosol. Okay, so when this kind of exchange occurs, this is known as passive diffusion. And why is this passive? Because it is moving down the concentration gradient, right? It is not moving against the concentration gradient. Okay, next we have facilitated diffusion. What is the difference between passive and facilitated? It means that other agents can enter the cell through specialized transmembrane carrier proteins. What are used over here? We have specialized transmembrane carrier proteins that are able to transport substances from outside to the inside. Okay, but still the concentration gradient remains same. It remains from a region of higher concentration to a region of lower concentration. But there are certain substances which cannot be transferred directly through the cell membrane. Why? Because they are not lipid soluble, and this cell membrane is made up of entirely lipids, so they need certain transporters which are known as transmembrane carrier proteins. Okay, the third one is active transport. What happens is that you basically have to transport the drug in the opposite concentration gradient, against the concentration gradient, from a region of low drug concentration to a region of high drug concentration, and this is saturable and it requires ATP. Why does it require ATP? Because we are transporting it against the concentration gradient. We are going against the rule, against the thumb rule. Okay, so that is why we need ATP, and ATP is being converted into ADP as you see over here. Next we have endocytosis and exocytosis. What happens over here is that it involves the engulfment of a drug by the cell membrane and transport into the cell by pinching of the drug filled vesicle. So what happens is that this drug molecule comes over here and it pinches as a vesicle right here, and then it moves and it is transported. Vitamin B12 is transported across the gut wall by endocytosis, whereas certain neurotransmitters are stored in intracellular vesicles in the nerve terminal and they are released by exocytosis. So whenever we have to release a drug, that is exocytosis, we have to take in a drug, that is endocytosis. Okay, and what is the example of the substance that is being transported? It is vitamin B12. Okay, so I hope this is clear. Next we move on to the factors that influence the absorption. Okay, so the first is pH. Whenever, see, acids have different pH and bases have different pH. Now we know that neutral pH is 7, and the substances which have a pH above 7, they are known as bases, and the substances which have a pH below 7, they are known as acids. Now what happens is that acidic drugs, they release a proton and they cause a charged anion to be formed. Okay, whenever acid releases a proton, it forms a positive H positive. Okay, and then it forms an anion, it forms a charged anion. Now what does weak bases do? Weak bases lose a proton, it produces an uncharged base. Whenever they lose a proton, so it produces uncharged base. So this is not that important regarding the your MCQ point of view, but you just need to know the concept. The lower the pK alpha of the drug, the more acidic it is. Now what is pK alpha? It is the measure of the strength of the compound with the proton. When we compare the strength of any material, any liquid with a proton, with the strength of the ionization of a proton, or the strength of capability of forming an acid, or capability of releasing an H positive ion, as according to this picture, so that is pK alpha. Okay, so when pK alpha is low, it means that the drug is more acidic. Why? Because it does not release protons. Okay, it keeps the protons with itself. Conversely, higher the pK alpha, the more basic the drug. Whenever the pK alpha is high, so the drug is basic. Distribution equilibrium is achieved when permeable form of drug achieves an equal concentration in all the body water spaces. Okay, so this is last point is not that important. Let's move on to the next. Blood flow to the absorption site. The intestines receive more blood flow than does the stomach. So absorption from the intestine is favored over the stomach. Okay. What happens is that intestine has got a wider network of blood capillaries. That is why absorption is being favored in the intestine as compared to the stomach. So this is another factor which influences absorption, that those drugs which are being absorbed in the stomach, they are less frequently absorbed, but those in the intestine, they are more absorbed because the intestine has got a huge capillary network. Okay. Total surface area available for absorption. This is another factor which influences the absorption of the drug. With a rich surface of the brush border containing microvillus. Intestine has a surface of 1,000 folds that of the stomach. Okay, so these 2 points compare the intestine with the stomach. First, the intestines have a greater blood supply. They have a vast blood supply, and they also have a vast network of microvillus and villus. Okay, so that is why the absorption increases. Next, we have the contact time at the absorption surface. If the drug moves through the GIT tract very quickly, as can happen with severe diarrhea, it is not well absorbed. Okay. Anything that delays the transport of the drug from stomach to the intestine delays the rate of absorption. Okay. The presence of food in the stomach both dilutes the drug and slows gastric emptying. Therefore, a drug taken with a meal is generally absorbed more slowly. Okay. Whenever we take a drug with the meal, what happens is that the drug is being diluted and it keeps slowly and slowly being absorbed with the meal. It is not absorbed like rapidly. Okay. And also when a person has diarrhea, so the food can move very rapidly within the GIT tract and he can lose the drugs which he has taken and they have not been absorbed in his body. Next, we have the expression of P-glycoprotein. What is P-glycoprotein? It is a transmembrane protein responsible for transporting. See, over here in facilitated diffusion, we studied that there are specialized transmembrane carrier proteins in order to facilitate the passage of larger molecules which cannot be easily passed through the cell membrane. So one of the examples is P-glycoprotein, which is used for the transport of substances that are drugs. Okay. See, this is P-glycoprotein. It forms a channel for ATP dependent pumping of the drugs from the cell. So what happens is that this is the drug concentration intracellularly and this is the drug concentration extracellularly. ATP releases its energy in the form of ADP and phosphate inorganic in order to move the drug from one place to another. This is expressed in tissues throughout the body, including the liver, kidney, placenta, intestine, brain, capillaries. Okay, so these are the locations in which this P-glycoprotein is being present. So you have to learn these. It pumps the drugs out of the cell. So it moves the drugs from intracellular to extracellular. Okay, and it reduces drug absorption. Alright. Facilitated diffusion, it can occur in the region of higher concentration to a region of lower concentration. So over here intracellularly, if the drug is more, if it's being toxic, then all of this substance, all of the drug is being transported to the extracellular environment. Okay, where we have the less concentration of drug. Okay. Next we have bioavailability. What is bioavailability? It is the rate to which an administered drug reaches the systemic circulation. It is a time, it is a percentage, it is a fraction of the substance which reaches the systemic circulation, that is our basic circulatory system. For example, if 100 mg of a drug is administered and 70 is absorbed unchanged, the bioavailability is 70%. Okay, so the drug which is absorbed upon the total number of drug which is taken, so wait, I'll write its formula. Bioavailability is the percentage. Okay, it is the amount of drug absorbed upon the amount of drug, total amount of drug taken, multiply by 100. So if the drug absorbed is 70 and the total amount of drug taken is 100, right, and we multiply it by 100 to get a percentage, what happens is that 70% of the drug is being absorbed, so the bioavailability is 70% or 0.7. Okay. It is useful for calculating drug dosages for non-intravenous routes of administration. See, in the intravenous routes of administration, as we studied earlier in the previous videos, if you haven't seen them, go watch them out. See, it has a maximum degree of control over the amount of drug delivered. Okay, so it is being delivered almost immediately and in full concentration. It is not being wasted, right? But in other routes, it can be wasted. Okay, so that is why we need to calculate the bioavailability. Why? Because the orally administered drugs, they undergo a first pass metabolism in which the drugs are being passed to the liver and they pass through the hepatic portal vein and then they are metabolized. So this thing actually affects the bioavailability a lot. That is why it is being calculated, that how much is actually being absorbed in our body. Now let's move on to the first pass of hepatic metabolism, which is like a big explanation. When the drug is absorbed from the GIT tract, it enters the portal circulation. How does it enter the portal circulation? Because when it is the drug is being absorbed from the stomach, it is transferred to the liver, then to the hepatic portal vein, and then it enters into the systemic circulation. If the drug is rapidly metabolized in the liver or the gut wall, the amount of unchanged drug entering the systemic circulation is decreased. Now what happens, we studied this earlier, the first line over here, that the drug may be biotransformed through metabolism by the liver. Okay, so the liver is used for metabolizing the drug. Okay, that is why we are saying here that if the liver metabolizes this drug, so the whole amount of drug cannot be absorbed, right? See, this is referred as the first pass metabolism. It limits the efficacy of oral medications. More than 90% of the nitroglycerin is cleared during the first pass metabolism. Hence, it is primarily administered via the sublingual, transdermal or the intravenous route. So, nitroglycerin, it is cleared during the first pass metabolism. It means that it is being, the liver, it degrades nitroglycerin, right? That is why it is transferred, it is given through the sublingual route. Okay, drugs with high first pass metabolism should be given in doses sufficient to ensure that active drugs reach the desired site of action. Next, we move on to the solubility. Now, very hydrophilic drugs, they are poorly absorbed. Why? Because they cannot cross the lipid rich cell membrane. See, these cell membranes, I told you, they are very rich in lipids. So the hydrophilic compounds, they actually do not dissolve in lipids, right? So that is why they cannot cross through it. They are insoluble in, these drugs which are lipophilic, they are insoluble in body fluids and they cannot gain access to the cells. For a drug to be absorbed, it must be largely lipophilic, yet have some solubility in aqueous solutions. So it needs to have both of the properties, that it needs to be lipophilic as well, which means that it can dissolve in lipids, and somehow it should dissolve in aqueous solutions too. That is why many drugs are weak acids and weak bases. Okay, because if they will be weak, they will not be able to perform one function very nicely. For example, if they are lipophilic, they will be extremely lipophilic and not hydrophilic at all. We don't want that. We want the drug to be both, to be compromising, to be adjustable in a lipophilic environment and also in a hydrophilic environment. Okay, that is why our drugs, they are weak acids and they can also be weak bases. Next, we move on to the next factor which influences bioavailability, that is chemical instability. Some of the drugs, they are unstable in the pH of gastric contents. See, the pH in the gastric tract will keep on changing. For example, in the stomach, it is acidic, whereas in the intestine, it becomes basic all of a sudden, right? So some of the drugs are unstable. For example, penicillin. Others such as insulin, they are destroyed by degradative enzymes. So when the drugs are being destroyed, the drugs are being metabolized, so automatically the bioavailability decreases, because the drug which we took into our body, the same effect will not be performed because all of that drug, it will be metabolized. Nature of the drug formulation. Drug absorption may be altered by factors unrelated to the chemistry of the drug. For example, particle size, salt form, crystal polymorphism, enteric coatings. Okay, so all of these factors determine how the drug is being made and what effects will it have. Next, we move on to the bioequivalence. What is the meaning of bioequivalent? The same dosage form, same active ingredient, same strength, same route of administration. These are the 4 main points that you need to learn. The first one, same dosage form. The second one, at the same strength. Number 2. Number 3, same route of administration, right? And number 4 is the same active ingredient. You need to learn all these points, and these drugs are called bioequivalent. Alright, so we are done till here. In the next video, we're gonna start with the drug distribution. So watch the next video as well. Till then, take care. If you have any queries, let me know. I'll be glad to solve them. Take care. Bye-bye.